Okay, we'll get going with this. I hope I got that set up right. It says live, so that's gotta mean it's working. And I always wait a second because sometimes it starts playing as I'm talking. What? Okay, any questions or concerns before I jump into today's material? I have seen a number of people already getting on the pack back and a handful of questions already getting populated in there, so good work. All right, quick reminder, Monday there is no class, so keep that in mind. All right, we left off with a phylogenetic tree showing bacteria and archaea in the eukaryotes. The next slide is not actually out of your book, although I left figure 112 up there. That's the picture in your text. This is a slight modification, one that I thought was a little bit better. There are, I feel like, a few issues with the one in the book where it's not exactly right. I've got, I think some of the viruses shifted over a little bit or something like that. I can't remember, but I'm like, I don't like the way that looks. So there are all sorts of examples like this image to get us thinking about microbes in the first place. There are all sorts of potentials that we may have at our fingertips to be able to visualize some of the unknown, if you will. And it really kind of just depends on the circumstances. Oh, wait a minute. As far as what we might be using, all right? So initially, we'll start off over here with just our eyes. There are numerous things that we're capable of visualizing without needing a microscope, all right? And even when we get into the realm that maybe a magnifier could be useful, we could still probably discern that. You can see a chicken egg probably in your fridge, maybe not with the cost of eggs, right? So we'll see, right? All those backyard ranchers are just laughing. They're like, it's all fine and good, right? I've got all the eggs I need, right? All right. So magnifiers, like just a little magnifying glass, a little hand lens, all right? That allows us to see some stuff a little bit better, right? And then we bump over here into our yellow bar, a conventional light microscope. That's what you're gonna see in the lab, all right? You're gonna use them a little bit, all right? In this course, I mean, microscopy is important. It is really, part of the whole process of understanding what you're dealing with, but it is a kind of process that's a little bit labor intensive at the start. If you've never dealt with any of it, it takes a little bit of time to stain stuff up and to find it under the microscope. After you've done it a handful of times, kind of just becomes like second nature, and you're able to do it really pretty easily, all right? So we don't have huge amounts of time to spend really getting highly skilled at microscopy. And we know most of the audience, probably that's not what you're really going to be doing in the first place. So you're going to get a little bit of experience, but not something that you're going to do all of the time, all right? If you were really working in a microbiology lab, the odds are every stinking day you're going to do some gram staining, all right, which is one of the key staining techniques that microbiologists use to differentiate between some of the microbes, right? You can't look at something and immediately know exactly what it is you're seeing, all right? I can't stain something up and be like, oh my gosh, it's E. coli, right? I've got maybe some hints, all right? Based off of color and shape, and maybe if I knew where it was coming from, right? I might have some hints, but typically you're not going to know that, all right? There are some specialized staining techniques. We'll talk about some of this in a bit, that maybe that's a little bit different, all right? But typically You get some hints, but you don't have a solid answer. All right. So we can see quite a bit of stuff, but when it comes to the microbes, you can't see them with the naked eye. You've got to have a microscope. And as we can see here, there are some things, maybe like a frog egg, that a regular light microscope could help to give us some more details with. But typically, we're thinking other smaller things. All right. Single-celled protozoal organisms like this little paramecium, a type of ciliate, plant and animal cells, maybe in biology. If you took a biology course, you looked at some of these things under the microscope, chromosomes in onion root tips, maybe, right? Yeast cells, we saw an image of some yeast just yesterday or Wednesday. It's always the day before. It's yesterday or tomorrow, right? But it's really not. All right. And then we get over here to our fuzzy little bacterium, right? These guys, and we've got a ruler bar down here showing some size to put it into perspective. Bacteria, very small, all right? And comparing them to animal or plant cells, eukaryotic cells, those types of cells are monsters compared to the bacteria, right? About 10 times the size. And there's also a heck of a lot more going on inside of a eukaryotic cell compared to a bacterium. You can see other stuff, right? And oftentimes, if you're looking at eukaryotic cells, if it's not in the process of replicating, the first thing that you're really going to notice is a big nucleus inside of that cell. Pretty easy to recognize, right? And if it's typically these things have been stained up, usually it's a purple kind of sphere or circle inside of that cell, right? When you look at the bacteria, you're not seeing that, right? And if we were maybe superimposing a bacterium on top of a eukaryotic cell, that bacteria might be about the size of the nucleus in a eukaryote, right? Really pretty little, right? I think, I mean, looking at some animal cells, looking at some protists, maybe swimming around, that stuff's pretty cool, all right? Looking at bacteria, maybe the first time you see it, maybe kind of cool. I think not so impressive, right? Little dots, and they just sit there, right? Very little. And really, if you don't know what you're looking at, you might mistake dust or something else as these organisms because they are very little, all right? Not super exciting, all right? Definitely the first time you see it, you're like, I finally found it, right? That's exciting, but then you look at them and you're just like, oh, purple circles, right? Purple dots, right? Or maybe pink ones, dependent, all right? So little, right, compared to eukaryotic cells, all right? But as I said before, similar stuff is happening, right? It's just very simple, if you will, inside of these little bacteria. As we look at this and we're seeing with the light microscope, bacteria are right at the end of what a regular light microscope is really able to allow us to witness in the first place. They are pretty small, all right? We will see as we move through here handfuls of microscopic images, some of them taken through a light microscope, but a lot of them we use electron microscopes because then we can blow it up considerably larger. All right, you can get finer details about the bacterial cell, or maybe even looking at the insides, dependent upon the types of electron microscopy that you might be using. All right, so you can see this overlapping quite a bit with the electron microscope, allowing us to visualize animal or bacterial cells and smaller stuff. We can see viruses, which you cannot see with regular light microscope. All right, they are way too small for that light microscope to be able to pick it up, right? In order to really see them, we need electron microscopy. And with that said, they are still really pretty small, right? It's sometimes kind of like a hunting for a needle in a haystack when you're trying to visualize that stuff. And then we move to the last bit, what the electron microscope isn't going to allow us to perceive. We might be able to see some proteins, but DNA and potential to visualize atoms, not capable with these types of microscopy. We need to employ some other techniques. And there's likely going to be some other advances that allow us to see things a little bit differently. I feel like we're on the cusp of some of that stuff. There are some techniques like flow cytometry that allows machines to go through and evaluate and count cells, like maybe a blood sample. And they have some of these that are linked up with special types of microscopy to visualize these cells as they are moving through there. And in a sample, there is the opportunity, maybe a blood sample, moving through there, counting individual cells. Abnormal cells can also be detected. And in one sample of blood, the potential to maybe detect a cancerous cell with flow cytometry and the microscopy linked with it is a potential. There are lots of advances that we're really kind of seeing. So the crystallography can allow us to see things, proteins, DNA, and other atoms or molecules as we move to that point. So it's the yellow guy, the light microscope that we're going to focus on really as we move through here, although we will talk a bit about some electron microscopy a little later. All right. When we think of this, there is some magnification with the lenses, and we'll show you a microscope. You'll see them in the lab, but we'll show some in lecture a little bit later, where there's multiple lenses that allow us to go from low power, not very magnified, to high power, as magnified as you could get. All right. And basically, after that, at about 100x, and there's your oculars that you're looking through and the objectives that are doing the main magnification. Typically, the oculars magnify 10 times, and then your respective objectives that are below there have another. So 1,000 magnification is typically the maximum that we see with regular light microscopes. You can't get really any better resolution, any clearer of an image if we try to ramp that magnification up anymore with these types of microscopes. So 1,000's it, and they are very fragile and sensitive lenses, especially at this magnification. We'll touch more on that later on. So these images down below are all electron microscopy images. They have been prepared and imaged with those techniques where high energy beams of electrons are being bombarded at a specimen, and in turn you get these images, right? So as I said before, we'll talk about some different types of those microscopy techniques a little later. We just want to show you some images of how these bacteria might appear, all right? So some cartoon pictures and then microscopic images. And again, remember, these are magnified considerably, all right? So the spherical forms, we call these caucus, a handful of them, 2 of them together, or many, we would say cocci, right? Circles, spheres, really, they're big balls, all right? Bacillus are cylinders, all right? We talk about bacillus as a type of genus of bacteria, but it's also a shape, all right? So used kind of interchangeably, all right? Vibrio are bacillus-like, but they're kind of a little bit different. And sometimes this stuff, if you're not really doing it all of the time, it's difficult to truly say, right? Am I looking at Vibrio or am I just looking at some bacillus, right? They're kind of curved, kind of comma shaped, right? And here, really kind of a poor image because they're all slapped on top of one another. And it looks like maybe this is just a little zoomed in compared to that. That's kind of how I feel about it, all right? They don't look great, but kind of curved, kind of comma shaped. Coccobacillus, these are sort of oddballs. They're little stubby bacillus, rounded, more oval, right? More oval than our coccus and shorter than the bacillus. So we kind of slap both names together, coccobacillus, all right? And sometimes you see some like that. Most of what we think about are cocci and bacillus as far as our lab stuff, all right? Moving over to the last two, these are kind of odd looking ones, sort of little spring guys, all right? A spirillum and a spirochete, they're curved more so than the vibrio, all right? And they have abilities really to kind of spiral through their environment. This a little more loose and this more tightly coiled, if you will, giving them a little bit of separation between the two, all right? So we'll think more about some of that as we progress through and start looking at all of these different types of organisms throughout the semester. So this image comes from kind of our backyard, from Yellowstone National Park, and truly all sorts of awesome things in Yellowstone. If you haven't had an opportunity to check it out, I recommend that you do. There are some pretty fascinating things, and a lot of the, you know, thermal pools, a lot of the features like that. We think about a lot of different types of microbes that are kind of setting the stage to see some of that in the first place. Some bacteria, but lots of archaea. And these, you know, before all grouped with the monarans and these oddball archaeas, we said were extreme bacteria before we separated them apart. Extreme, like being able to thrive in boiling hot water, right? None of us want to hop in there. You shouldn't, right? But those organisms, that's what they find suitable, right? They thrive in those types of habitats. And there's all sorts of other opportunities, all right? So this, really a lot of this, we could see the archaea and the coloring within here. There are sulfur molecules that, you know, they are able to pull in and like respire on. And that's where we're seeing some of the yellow is the sulfur atoms that ultimately get manipulated into some type of a molecule, like hydrogen sulfide, stinky stuff, rotten egg sort of smell. And bacteria are pretty good at dealing with some of that. You'll see a little of it in the lab, right? Kind of interesting. Yellowstone opens up the avenue for all sorts of ideas with molecular biology. And I'm going to put the brakes on that. We will talk about that a little bit later, all right? But a lot of what we think about with being able to manipulate stuff to do PCR reactions or polymerase chain reactions are the result of research that happened, maybe not necessarily at this specific thermal pool, but in Yellowstone, all right? I'd like to imagine maybe it was some water sampled right from there. This, I'd also like to say came out of Yellowstone, but it's not. This is from Antarctica. Diatoms are crazy. This image, I think, is kind of impressive. There are oftentimes images of them that you're just like this. It's like some sort of crazy psychedelic poster that you feel like you could just watch for an hour, right? Weird, very weird. And if this was a droplet of water, we would see them moving around, doing all sorts of crazy things, right? Journey to the microcosmos, I'm sure, has some diatoms that you can witness. Did anybody check out that video that was posted on there? Not a soul in here. Whoa, holy cow. That's not connected. Better be careful. All of a sudden, I ripped the room apart. they'll ship me back up to biology. They're like, you can't play down here in Rendezvous, right? The destroyer. All right, so diatoms, these are odd, all right, and potentially used not the living forms, but the dead carcasses of diatoms are used in some organic gardening. They are what we call diatomaceous earth. And they're like little shards of glass that you can spread around your tomatoes or whatever. And if an insect decides it wants to come check out your tomato plants, crawls through the diatomaceous earth, and those little shards of glass get into the exoskeleton and basically cut it up, and the bug dies. All right, kind of crazy stuff. All right, so that's like what we're looking at here, seeing these cells, the cell walls of them, algae, has lots of silica, which is what glass ultimately is composed of. Really pretty cool. Oh man, Giardia. Look at my watch. How much time do I have? Giardia, I think, is so awesome. This we're shifting over to think about a type of eukaryote, the diatomaceous earth, or the diatoms. They're eukaryotes as well. But this is something that is a human pathogen, and definitely in Idaho, it's our #1 parasitic infection, right? Probably the whole US, it's the top, all right? It's something that we get by ingesting water that's been contaminated, oftentimes camping, hiking, backpacking, stuff like that, or maybe you drink some water that you think looks pristine, bubbling right up. I found some sort of spring, I'm gonna drink this water. Maybe not the wisest choice, all right? If you filtered it, or if you boiled it and cooled it, that's a different story. But if you just drank it right from the source, maybe not a good idea, all right? It's also something that takes a bit of time. So people typically don't put it together that it was the camping trip they took a month back that's the reason why they're crapping themselves to death currently, right? Oftentimes you get a bit of intestinal discomfort, and the first thing you think of is, where did I eat last, right? What restaurant did I go to, right? And you want to blame that. Most foodborne illnesses happen in your own house, not from some restaurant, even though you want to blame somebody, right? So that's what's on your radar. Typically, things like that run their course, right? A day or two of that, and you feel good to go. But Giardia is not like that, right? Days later, you're still running to the toilet, wondering what's up, and that's probably when you go to the doctor, right? There are good amounts of individuals that consume some of this stuff and see no ill effects, all right? But if you get enough of it, I think you're probably going to feel the ill effects, all right? So Giardia lamblia, these are flagellated protozoal organisms. They've got these humongous suckers on the underside, and they like to stick to your intestinal cells and potentially filling up all sorts of space, all right? I'm not going to go through more details with that right now, because we're going to see Giardia again. Although I want to, I'm like, oh man, I could talk about these cute little guys all day long, all right? But I won't, all right? And potentially, some of you maybe have dealt with some of this in your life. Maybe you don't know, right? This next image, also eukaryotic. These are some yeast cells, not the Saccharomyces that we started with when we were looking at the bread stuff. All right, Candida albicans is a type of yeast that's a pathogenic form. And this is something that really you have lower levels of it as just kind of a normal population. And it's when things happen that sets the stage for other cells like bacteria to get depleted that lends to the yeast cells proliferating. All right. So it's a culprit for vaginal yeast infections, and it can also cause oral thrush. Same idea, just different areas of the body. All right. There are definitely potentials where, with thrush, it's a good one or a bad one for little kids as they're, fresh hatchlings, infants may potentially get this, and their body is really just figuring out the world, right? They're kind of prone to getting all sorts of different things, and thrush is kind of a common childhood infection, right? If your normal flora gets out of whack, that's when we think about other potentials. And as an adult, yeast infections, whether it's vaginal or oral, or potentially it could be a systemic sort of infection. If you're severely immunocompromised, it may make its way into your circulation and spread throughout your body. And then you're in some seriously poor shape. Not that you probably weren't already, all right? So they have the potential to cause some issues, all right? Typically, when we're thinking this, some discomfort, there are, it's relatively common, so much so that you really don't necessarily need to go to the clinic to get treatment. You can go to the supermarket and get products that help to deal with this, all right? So that lets you know that it is a relatively common situation, right? Oftentimes with the vaginal situation is the culprit is antibiotics that you might be taking for something that ends up impacting your good bacteria, what we call the normal flora, and that lends to some of these issues. So consumption of some probiotics, eating some yogurt, if you've been prescribed antibiotics, definitely helps to keep these situations in check so that you don't develop yeast infections. So there are a handful of these yeast cells within here, all kind of grouped up together. You can also see the nuclei in them. They are eukaryotic. The stain with some fungi is a little bit different than staining like some of our own cells. They also have a thick cell wall, so it all looks nice and blue, and then little darker blue bits inside that's the nucleus. That's what we're seeing throughout. There's some other stuff as they replicate where they kind of push out. We showed those bread yeasts that replicate by budding. These do the same thing, but you can also see some sort of pieces that emerge out of them that we say are pseudo-hyphae. Molds, fungi like that, have big filaments that we call hyphae. The yeast cells here, it's not really a filament, but under the microscope, it sort of looks like that. And if you're looking at Candida or you're questioning it, seeing some of the pseudo-hyphae might clue you in that that's really who you're seeing, all right? This pile of oranges is full of another type of fungus, all right? This is some mold, all right? The fuzzy filaments, the hyphae, or all of it together, we would call mycelium, all right? Individual threads, hyphae, and all of it mycelium, all right? So that's all of this stuff. Lots of times seeing either the darker or the lighter bits on there might be the spores that the fungi is producing that would be released out, settling back down and growing as more hyphae, right? So seeing them under the microscope, you see these kind of thread-like structures, and then the fruiting body where the spores will be there's a lot of variation with it, and we will show you some of these images as we move into the next chunk and start thinking some of the diversity. But it kind of, lots of them kind of remind me of a dandelion as it's ready to go out to seed, and it's all the, you know, the big bit on there that you could blow, right? That's what a lot of the fungal spores look like, and kind of the same idea. They would move and settle back down. So they can be seen with the naked eye, but also higher detail, like the dandelion-like stuff, if we were to see it under the microscope. All right. And also, you know, some pretty interesting stuff. All right. Fungi is impressive. All right. And we will touch base on a handful of those that can cause some pretty serious disease. These next ones, you know, all sorts of parasites. I'm just like, oh, I love them so much. This is another one that I'm just like, oh, geez. Another all day long lecture about tapeworms and guinea worms. Shoot. All right. These are two completely different types of worms. Our tapeworm right here is a flatworm. All right. Looks like a big piece of tape. All right. Potentially a giant piece of tape. These guys can be meters long, all right, upwards of 10 meters. That's impressive. You know, you got a friend, right? If you've got something like that going on, right? Back in the day, and maybe even still today, some people would take it upon themselves to deliberately infect themselves with some tapeworm eggs to in turn drop the pounds, right? Oh, I'll lose some weight. I'm going to give myself worms. Terrible idea. All right. And truly, as your body's being bombarded with a parasitic infection, you kind of oftentimes your body feels like I'm starving to death, because you kind of are, right? Maybe you are losing some weight, but you think you're starving to death. And when your body is in starvation mode, anything that you consume, you want to store as fat. So you're really doing yourself a disservice, right? But you never know. All right. So the eggs are really little, all right? But the adult stage can be gigantic, dependent upon the respective type of worm. Taenia saginata is the beef tapeworm. There are a couple of others that we think about impacting people. There are handfuls of them that can get all sorts of other things, but people, there's pork and beef, and there's some that you get from fish that give you some tapeworm infections. There are other flatworms that we call flukes. And then the other one with our little matchstick, the guinea worm, is a type of nematode. That's a roundworm. They're a little bit different, right? They're a lot different, really, all right? So with that, we will touch base with some of this a little later as well. I'm not going to spout off all sorts of details right now, even though I want to, all right? So here, what we're looking at on this individual, this is an ankle. Here's the Achilles tendon. That's gotta hurt, right? This worm, and I will tell you real quick, the flatworms almost always are hermaphrodites. One worm with both sex organs. It only takes one, all right? You don't get infected with this tapeworm and all of a sudden you've got millions of tapeworms. You've got, if you ingested one egg, or if you ingested poorly cooked meat, because with the tapeworms, typically it's not the eggs that we eat, it's the juvenile stage that's encysted in the muscles of some animal, like a cow, if we're talking the beef tapeworm. So you eat a steak that's not cooked thoroughly, and you potentially become infected, right? And with that, it grows up to the adult stage in your intestines. And if all you consumed was one cyst, all you get is one worm. And that's all you need, right? And that worm, as it makes its way to kind of maturity, it's constantly producing little segments that we call proglottids. And each segment has both sex organs they fertilize, they get full of eggs, and they either rupture and release the eggs out that you poop out, or they break off as little segments, which might be your first hint you got worms, as you have a segment of tapeworms that you defecate out, right? And that in turn sets the stage for something to consume it, like a cow, that in turn insists in its musculature, and the life cycle will be complete, all right? So one cyst that we ingest gives us a worm with the potentials to produce millions of eggs, but you don't get millions of worms inside of you, all right? When it comes to the worms, we don't think of them as proliferating inside of us like that, all right? So I'm telling you that because next door are nematodes. These worms are what we call dioecious, separate sexes, instead of the hermaphrodites or monoecious forms, all right? So the female is what's twisted around this matchstick here. After you become infected with these, and we typically get infected with the guinea worm, and it's very rare. Definitely don't see it here. The guinea worm infections, you drink an aquatic invertebrate. We call it a copepod. It's a juvenile stage of something very, very small, nearly microscopic, all right? It gets consumed with some maybe sort of not the best water, right? And in turn, the worm is released, and again, separate sexes. Fertilization happens. The male dies shortly after copulation. The female grows bigger. It takes almost a year for this process to truly complete, migrating toward the extremities, and a big ulcer forms, typically on the foot, all right? With something like that, my thought is, that's gotta hurt, right? Probably quite a bit. And if it hurts, you probably would like to soak it in some water. That's what that worm would like you to do. Put your foot in some water, and I'm gonna poke through, and I'm gonna lay a bunch of eggs. All right, that's what happens, right? And then the cycle is complete for that, right? The copepod eats the egg, you ingest that, done deal, all right? So the matchstick is a mechanism to slowly, kind of methodically twist it out of your body. The female is about 1 meter long. It's not like the tapeworm, but 1 meter is still a pretty good sized worm, right? So slowly, it takes maybe a couple of weeks to truly rid yourself of this worm, all right? That's kind of disturbing, right? And if you try too quickly breaking it off, then you've got some other reactions that occur with this dead worm, and your body's trying to mount responses against it. You want to be very careful as you deal with that. This is something that has plagued humanity since antiquity, right? There are all sorts of accounts of guinea worms and details, all right? There are images from ancient times of these individuals, the medicine man or shaman or whomever it might be, wherever you're at, images of this stick and worm, which is really what they think, what is it, the medical insignia with the snakes around the staff. What's that called? Caduceus? I don't know. I don't know if that's right. Some of you people in here, that's what you're going into. Although those serpents look snake-like serpents, these worms, we're thinking serpents as well. The thought is that's what it's all about. With these snakes twisted around the staff. Are you looking it up right now? Asclepius? Okay. I could just tell. I'm like, someone's looking it up. All right. Crazy. All right, to think that maybe that's really where it all comes from. The doctor will twist your worms out, right? That's what it's about. This is something you know, lots and lots of infectious disease is just something that we flat out deal with, right? There are a few things that we could say, yeah, we've eradicated, like smallpox. We don't think about it, right? This, the guinea worm, we are so close to saying we're done with it, right? A very few infections, right, are occurring in people. We're almost at the point of saying guinea worms aren't a problem for us, but they're still going to be a problem because these worms, they don't have to just infect us. They can infect other things as well. And now a bigger problem, it's not people that are the problem. Now it's dogs. And the guinea worm in dogs is becoming a bigger situation. Dogs run in there and drink a bunch of water. And in turn, they can become infected. And now we've got, instead of a human issue, now it's a veterinarian issue, right? And similar sort of problem. Kind of cool though. All right, and this we have seen maybe, although we are looking at a different type of SARS. This is coronavirus. It's a close cousin, right? SARS-CoV-2, similar as what our pandemic has brought us in the first place. And both of these, electron microscopy, because you couldn't see these viruses without it, all right? And this one's not really the best looking one in the first place. Over here is Ebola, very different looking type of virus. As far as morphologies go, there's a lot of variation with some of that stuff. Virus have a protein shell that we call a capsid that might be kind of circular, or other images that we will see, or potentially cylindrical or long thread-like, all right? Some kind of icosahedral, really sort of cool looking. Depends on situations when we're thinking virus, both of these human viruses, but there are others infecting plants or infecting bacteria, all sorts of different details with that, and we'll touch base on a little of that as we progress through. Okay, and lastly, I knew I was getting close to the end. Here we've got a virologist looking for influenza A virus causes avian flu, bird flu, and this is something, you know, it's an older image, but this is something we're dealing with right now. I recognized a couple of days ago as I was scrolling through on my phone, eagle in, I think it was Virginia, that they had tested that had avian flu, which is also why the eggs are so expensive, so they say, right? Problems, all right? All right, I'm gonna quit with that. Littlest bit early. Questions, concerns? Farewell. I will see you all Wednesday.